Good afternoon. Once again, welcome to this panel discussion and Q&A on the science behind the COVID-19 vaccines and other non-pharmaceutical interventions aimed at mitigating the spread of the virus. Before we begin, I'd like to introduce our panelists. With us are Professors Carolina Herrera, Stu Feinstein, Scott Grafton, Joanne Candela, and Chuck Samuel. So let's jump right in, starting with a question about the effectiveness of the COVID-19 vaccines. Professor Grafton, what degree of protection do the vaccines provide in terms of symptoms and of severity of disease, and also in terms of disease transmission?
Well, thanks, Andrea, and thanks to the audience for actually tuning in and learning some more about this. This is a really fast-moving area. Just to give you a sense, we had a New England Journal article updating a lot of this information just 2 days ago. So we're all trying to keep abreast of how good these vaccines really are. And I think the news overall is very, very good. If we look at the two approved vaccines we have right now from Pfizer and Moderna, those are the two RNA-based ones. You know, they've been out now for quite a while. We can talk about those. And we also have this brand new one from Johnson & Johnson that's a DNA-based vaccine, and that's being reviewed by the FDA tomorrow, and the evidence for that one is already out in the public domain, so you can look at how effective that is. So without getting really into the weeds and the specific numbers, I think we can make some broad generalizations that help you keep in mind how well these work. And also keep in mind that for the FDA to approve a vaccine, the maker has to show that it helps or prevents disease 50% of the time. So that's a pretty low bar, okay? That's what's required. Now, all three of the vaccines that are out there, the two RNA and the new DNA one, are all greater than 90% effective at protecting you from hospitalization, severe disease, and death. So they're way above that low bar that's set by the FDA. To have a vaccine be 90% effective or better is absolutely phenomenal. Now, it turns out those two RNA ones that we have out in the public right now are also 90% effective at reducing or preventing mild and moderate illness. The J&J one, Johnson & Johnson DNA vaccine that's being reviewed tomorrow, that's preventing disease, mild to moderate disease in about 66% of people. So it's not quite as effective for more mild disease. However, there are a lot of caveats to comparing them directly. Different populations, different virus strains. The J&J one, you use only one dose, where the two that are out there right now, you use 2 doses. So there are a lot of reasons why the efficacy for milder disease might be different between the vaccines, and that's something we need to flesh out. Now, in terms of, you asked, Andrea asked about adverse effects. We now have 45 million people in the US that have been vaccinated, and so we know a lot about adverse effects. Mostly it's just injection site pain, and then some people get flu-like illnesses that are, muscle pains, joint aches, a little bit of fever, things like that last for a day or two at most. It's more likely with the RNA vaccines than with the DNA vaccine, and it's more likely in young people than old people. Now, one of the questions I get asked all the time is, if I didn't feel anything when I got the vaccine, does that mean it doesn't work? And if you go back and you look at the FDA's data that the manufacturers provided, there's no relationship between efficacy, when it works or not, and who did or didn't get symptoms. So don't panic if you get the vaccine and your arm doesn't hurt a lot compared to a friend of yours. One other thing that came up is anaphylaxis. This severe allergic reaction. There were some reports of these early on, and this was of great concern. We now have enough data out to know that it's actually very rare with the two RNA vaccines. If you look at Santa Barbara County, If you look at all 450,000 people in Santa Barbara, if we vaccinated all of them, 2, maybe 3 people will have anaphylaxis at most. That's the risk. Now you can compare that to allergic reactions of other kinds, where about 9,000 people in Santa Barbara County will have, severe allergies to bees or peanuts or other things. So it's, we're monitoring for that, but it's not as great of a concern as people originally thought. I thought I'd finish up with just 2 other quick points. One is some data just came out of Israel that's really exciting and important. This is a study of 1.2 million people, half of whom were vaccinated and half of whom were not, with the Pfizer vaccine, the 2 doses. And they basically reiterate what the original FDA data showed, but it also shows something else that's really important, which is if you look at people who got the vaccine and you measure whether or not they're carrying the virus a week after their second dose versus people who didn't get the vaccine, there's about a 90% reduction in them being infected. And that includes asymptomatic infections. And that's always been a big worry, right? This is a virus that transmits in a lot of people asymptomatically. So if you can show 90% protection, even in asymptomatic cases, that's a huge win for all of us. And one other point, the Israel data comes, it was acquired at a time when that UK variant was quite prevalent in Israel. So that's good news as well. So it seems to be working against that variant. One last point, and that is new data just came out on US nursing homes and how many people are sick in our nursing homes. And you can see a really precipitous decline in the number of COVID cases in our nursing homes that's emerged since vaccination started. And recall, our elderly were some of the first to get the vaccines. And so I think it's early news, but it's really promising that things are starting to kick in with these vaccines in our population.
And so obviously we've not had enough time for long-term studies of the vaccine safety. So what then can we know about potential long-term impacts or side effects? Professor Arias, and I apologize for misspeaking earlier.
No problem, Andrea. Thank you. And yeah, that is a pretty good question. And as Scott was just mentioning, the majority of the adverse effect that we're seeing in the people vaccinated occur within the first to a couple of days following the vaccine. And these are, as we said, headaches, malaise, minor or mild adverse effects. And of course, we have, as Scott mentioned, the anaphylaxis. One thing I can tell you is that the safety of these vaccines is under tight scrutiny by different entities. And the CDC, the FDA, and other institutions are following closely these 45 million people that have been vaccinated. And so far, it is pretty encouraging that we're not seeing any of these, let's call them delayed adverse effects. We have information now for over 8 months when the first doses of these vaccines were used in clinical trials. So it's not that we have just a couple of weeks or a couple of months to evaluate these adverse effects, but we actually have several months that we can follow them. And with other vaccines, there are some reactions that can be delayed. For example, Guillain-Barré with some other types of vaccines. But what is really, really encouraging is that for all these months that we have been following people since the clinical trials, since the first doses were given, we're really not seeing the reports of these adverse effects, which is pretty encouraging. And it's looking like these vaccines are safe and are safer and safer as the time goes by and as more people get vaccinated. So again, this is something that is under constant monitoring and under constant scrutiny by different institutions. So far the data is pretty good.
So before we go any further, Professor Samuel, can you explain for us how the Pfizer and Moderna mRNA vaccines, how they work and how they differ from live attenuated vaccines?
Well, thanks, Andrea. Yes, the mRNA vaccines or the RNA vaccines that Scott referred to do represent a new approach to immunization. And you're spot on correct. By contrast, live attenuated vaccines have a very long history, and we're more familiar with them. And there have been some great vaccines that are live attenuated vaccines. Measles, mumps, rubella, polio, smallpox. These were all live attenuated vaccines. Smallpox has been eradicated. Polio is nearing eradication. Measles, for the most part in the US, has been eradicated. So immunization and vaccination can work marvelously well. What a live attenuated vaccine is, as it suggests, it's a live virus. So when you're immunized, the virus actually replicates, but it's attenuated, it's avirulent. So it's a weakened version of the disease-causing virus, and it's weakened by mutation. Usually those viruses have a large number of mutations, and to generate and characterize such a vaccine virus takes a long period of time. So it takes, can take years to develop a live attenuated virus that's the whole virus antigen. Now, by contrast, the RNA vaccines, the two that are approved that have been referred to for the coronavirus, so CoV-2, that's the cause of the COVID disease, that's the pandemic that we're dealing with. These coronaviruses are large viruses. So in terms of genetic information, it's in the order of 30,000 nucleotides, 30,000 building blocks. And one of the genes of the coronavirus specifies a protein that's called spike, that's on the surface of the virus particle. And usually in viral vaccines, the protection that's generated by immunization is against a surface protein component. And so the RNA vaccine takes about 10% of the coronavirus's genome and expresses just that to make the spike protein that then gives the protection that triggers the immune response. In the Moderna vaccine that had been initially codenamed mRNA 1273, it was named that because the spike protein is 1,273 amino acids, the building block of proteins, amino acids. And so, as I say, this is about 10% of the genome. So what's done with an mRNA vaccine then is the mRNA is made, and this can be done very readily and very quickly with our tools of molecular biology. And so the mRNA that codes for the spike protein, the protein that's going to be the antigen that triggers the immune response that gives us protection for when we then are infected with the disease-causing virus. This mRNA then is loaded into small lipid bags. And when we're vaccinated, immunized, that mRNA is introduced into us, and our bodies then make spike protein that is the antigen that triggers the immune response. So there are two of these mRNA vaccines. One is Moderna. It's actually Moderna NIH because it was co-developed by the National Institutes of Health and the Moderna biotech company. The other is the Pfizer vaccine, or Pfizer BioNTech. It was co-developed by those two groups. The two mRNA vaccines use a very similar strategy. It's just this small part of the virus's genetic information that's used, but there are subtle differences in the form of the spike proteins that's expressed. And to achieve success, not of one, but of two of these vaccines that work just marvelously well, is a real, in less than a year, is a real scientific triumph. So that's That's where we're at with the true approved RNA vaccines.
So yeah, it was only a year ago that the pandemic was really taking hold. How is it possible for these vaccines to be developed so quickly when the process usually takes years? Professor Feinstein, can you talk about that?
Sure, Andrea. This is actually, it's actually a nice story. I'll tell you, the way we got the vaccine so quickly has a lot to do with history. And so let me tell you that story. For the past, say, 50 years or so, the National Institutes of Health, the NIH, and many other funding organizations have made major investments in basic biomedical research, asking two basic questions. First question is, how do cells work, including cells of our immune system? And then at a more probing mechanistic level, how do the biomolecules in our cells make them work? And these many decades of work have given us two things. First, we have a very detailed understanding of how cells work. And second, we have a huge toolbox, an arsenal of biomolecules such as DNA polymerase, or reverse transcriptase, or messenger RNA, and thousands more that we know how they work, and we know how they work well enough that we can make them do things for us in test tubes. Okay, so mixing and matching some of these various biomolecules in clever and creative ways enabled us to build these new COVID vaccines that Chuck just described to you much faster and much simpler and much less labor intensive than using traditional vaccine production methods, such as he spoke about the attenuated viruses. So it should also be mentioned that these mRNA vaccines didn't just come out of the ether. People have been working on them for, say, a decade or more to optimize their effectiveness. So if you take all the parts of that story I just told you together, everything was really beautifully set up at this particular time in history for us to take advantage of all this knowledge and implement it so that we could make these vaccines. So in the final analysis, the world really reaped the rewards for very wise investment in the basic sciences over the past few decades. And I'll close with just an analogy. In a way, it's sort of the biochemistry version of what's happened in information technology over the past decade or two. We have all this incredibly powerful hardware and all this incredibly powerful software, and we're largely limited by just how clever and creative can we be in mixing and matching the parts to do things that we want to do. And so that's really the story of how the vaccine was generated so quickly. All the players in the cast were there and ready to go, and it was just a matter of writing the text of the play.
Wow. And so when we're talking about the process of developing the vaccines, obviously they involve significant clinical trials with large numbers of people. Can we talk about those clinical trials, and also have the clinical trials included people of color, and do we know they work equally well in all groups? Professor Arias?
Sure, Andrea, that is a critical question. So all of these vaccines have been evaluated for safety and efficacy, the points that we have discussed before, in major clinical trials that have included tens of thousands of people. We also know that, unfortunately, COVID-19 has disproportionately affected our communities of color. And for that reason, these groups should be included in these clinical trials. And they have been recruited and they have been included for the Moderna, Pfizer, and Johnson & Johnson trials, which are the ones that we have information about. Anywhere between 20 to 38% of participants were from different races and different ethnicities. We know, for example, that Black and African Americans have corresponded to about 10 to 17% of the people that were included in these trials. We know that Hispanics and Latinx go anywhere between 20 to 45% of the groups that are represented in these phase 3 clinical trials. And we know that our Asian populations are represented between 3.5 to 4.7%. So, and we also know that the reports are that the vaccines are effective in these groups as well. So this is actually a change from other clinical trials that have occurred in the past. And there is really a huge effort by these companies and by other institutes to try to recruit as many people of different races and ethnicities that are representing the communities that are affected by these particular diseases. So it has been a good effort, and we do have some representation.
Yeah, really important. And what about children? How about clinical trials involving children? And when might we expect vaccinations for them?
Right. So the vaccines have been so far evaluated in participants 18 years of age and older. So we are missing information about the efficacy and, again, the safety of these vaccines in younger populations. And it's going to be crucial for us to understand how effective these vaccines are in this group. It's going to be important for us to vaccinate them and to include them. Right now, Moderna finished their recruitment for one of their trials in teenagers. I think they are between 12 to 15 years of age, or 12 to 16 years of age. And they are opening another one for kids, for children, younger children that are between 6 to 11 years old. It's very likely that we're going to know more about these clinical trials at probably around the summertime. And I know that Moderna, Pfizer and Johnson & Johnson, other of these companies are also starting some clinical trials. So people are again, companies are making the efforts to include children. When is that going to happen? Again, the timeline depends on what we see in the trials, and it's around the summer.
So let's turn the conversation to the variant strains of the virus. How do these variants come about? Are they a natural occurrence? And what can you tell us about the new Cal-20 variant?
Sure, Andrea. I can start with the first part of where did these variants come from that we are hearing about so much? And I can tell you that this is it's not unexpected. It's the natural evolution of the virus. As the virus replicates its genetic material, sometimes it incorporates some errors that result in mutations. As these mutations accumulate, we have what we know as variants. These variants may have, or these mutations may have, some effects on how the virus is transmitted, or in the virulence, or in the way that they cause disease. If there is an advantage, some viruses are going to be selected versus others, and they are going to transmit more efficiently, and they're going to accumulate in the population. And that's what we have seen with some of these variants that we have been hearing about. We know, for example, that the one in the UK, the B.1.1.7, is more transmissible. And we have seen the shift in the prevalence of these strains from November till now. And some of those have become the dominant strains in the population. So again, this is something that is expected, and is something that every RNA virus does. This accumulation of mutations is nothing rare that completely took us by surprise. And other mutations will accumulate, and other variants will arise.
So Carolina, would
Yes.
you say then that that's a reason to get immunized, and soon, before the variants arise and could have an opportunity to spread?
Yes, absolutely. I think that these vaccines are going to be one of our best lines of defenses. And again, they're going to offer protection even to other variants, even if it's not exactly the variant that was used for the development of the vaccine. We have seen already, as Scott was mentioning earlier, that they are offering protection from some of these variants. And they may also offer protection for some of the variants that could arise in the future. So yes, I think it is important not to wait and get the vaccine if you are able to, as soon as possible. And we can talk about that in a moment as well. But as you were mentioning, Chuck, these mRNA vaccines are very versatile, right? And so if there are strains that are arising where we see that the vaccine maybe have a little bit less efficacy than what we are seeing for other strains, they can be easily changed, and they can be easily manufactured so that we can have a booster that could offer protection to us, to some of these new strains and to some of the arising strains. So I think it is important to, again, as we said, just get a vaccine as soon as you're able to. And if there is a need in the distant future to get a booster against some of these variants, it can be done as well.
So, so how would that work? You know, with the boosters or repeated vaccinations. So someone gets the vaccinations, gets their doses now. Maybe 4 months down the road, there's a variant that requires a different or a booster, and then it's just a matter of going back to the doctor and getting that. And those vaccines will just be made available.
Yes, that's the plan. And Moderna and Pfizer have actually been talking, and I think Moderna has started the development of some of these potential boosters. We'll see again in the future if there is the need for boosters against new variants or boosters that are going to increase your antibody response. So all of this is still in development, but so far what we have seen is that the vaccine that is available right now, it is good, it is effective, and it is offering protection.
You know, even if we do need a booster and the vaccine needs to be tweaked going forward in the future, I mean, this is not a new situation of having multiple serotypes and genotypes. Polio, for example, there are 3 different polio viruses that we're immunized against. Or HPV, the Gardasil vaccine, that was 4 initially, and now I think it's up, maybe Scott knows, it's up to 9. So the idea of multiple serotypes, you know, we've faced before and certainly solved.
And that's not a whole lot different. We get a new flu vaccine every year because you get changes in the virus. Just a couple things to add on to what Carolina said. And I just want to emphasize the point that Scott made earlier on, that Israeli study that showed such good efficacy. For the most part, that was the B117 variant that originated, that we first saw in the United Kingdom. So in this case, that says the Pfizer vaccine was very, very effective against that variant, even though that variant does have increased transmissibility. So I think that at this point, now variants could arise that are going to be problematic for the vaccine. That's certainly possible. But most of the ones most everything I've read about the current vaccines that are out there says that yes, some of the variants have reduced reactivity with the antibodies, but it is still very much well within the levels. The remaining activity is very capable of protecting you. Okay, so we have not had everyone's worst nightmare happen yet. And as for the Cal-20C that you mentioned, Andrea, I think the analysis on that one is still pretty early and the data is pretty preliminary. I think much more data and better data will be coming along in a matter of weeks, not months. And so I think people should just sit tight on that and let's see where the science takes us, because there's a lot of stuff coming out every day. There's more stuff coming out, but it all needs to be really read with a critical eye to glean from it what we should really be about it.
So taking all of this into account, it seems that, you know, if not the virus itself, then certainly the variants suggest that we need to continue maintaining our effective COVID-19 behavior, which includes wearing our face coverings and physical distancing from people outside our households. So Professor Incandela, can you talk about aerosols and ventilation in the context of why face coverings and perhaps even double masking continue to be critical to mitigating the spread of the virus and these variants that are arising?
Well, yeah, certainly. I'd be glad to. I think the main thing to keep in mind is that we have kind of a battle that we're in with this virus, and it's not just a single virus, as you've heard. There are variants, and there are some great weapons that we have adapted now that are kind of, you know, capable of really addressing this in a very definitive way. But but even before there were vaccines, we knew that these mitigation steps that are often recommended, you know, keeping 6-foot distance, wearing masks, being careful to stay in well-ventilated spaces and so forth. We know that these are very effective. And if everyone had adopted them very religiously, you know, things might have been very different. But it's difficult to get everyone to do that, and we know that. In any case, it's important to continue to wear masks, keep distance, because we have the issue of how long it's going to take, for instance, just to get everyone vaccinated. There are some questions about when people have been vaccinated. For instance, after a single dose, they can still be transmitting the virus and get infected at some level. I'll leave that to the medical experts here to explain more detail. I don't know what will happen after the second dose. I think there's still studies underway, but even taking that into account, there is the possibility of variants. Until we really knock this out, we're not sure what we're going to be facing. Now, you asked about aerosols, and I just wanted to mention that I think for a lot of people, we heard in the very beginning about how there was a worry about basically, let's think about just how this is transmitted. People produce droplets of fluids from their mouths, from their nose, whatever, sneezing, coughing, talking, and these, if you touch surfaces that have them and then touch your face, you can get it through your nose, your mouth, your eyes. You can get it directly if somebody coughs in your face. We all kind of understand this projectile model of droplets that contain the virus. What I think was underestimated were the small droplets and the impacts they have. And when you get down to about 1/10,000th of an inch, which is very small, you're talking about what are called aerosols, just very small droplets. And the reason they're a serious concern is that they don't fall to the ground, they don't fall to surfaces, they don't get cleaned up, they float and they can accumulate. And the virus can remain viable in aerosols for several hours. So indoors, in winter, in a room that's not well ventilated, if you have multiple people and one person is infected, their exhalations, their aerosols will eventually accumulate and could reach levels that are infectious, even for people quite far away from them in those rooms. And this is what leads to this winter effect of people going indoors and getting infected. And this is why I think people are now recommending that people wear better masks, much more effective at filtering. But I like to say that what you should always do is be aware of your circumstances. Make sure there's good ventilation. If there's not, get outside or don't accumulate with other people. Distance may or may not help you. And even outdoors, you have to think. If you're running behind somebody who else is running, who's also running and breathing hard, they're actually creating a wake of spray that's just pouring all over you. So be aware of where you are. Wear a mask, if possible. Wear 2 masks, and you'll be helping tremendously to fight this battle.
In terms of wearing 2 masks, what is the best? What can you say about, or guidance can you offer about kind of the double masking practice?
So here, the issue with masks, a lot of this is mechanics, and the people wonder why I'm on here with all of these great biological and medical experts. I'm the physicist, so I guess I'm here for the mechanics part of the story. A lot of this is mechanics. The mask, there's 2 components to a good impact of a mask. One is its intrinsic filtering capability. That's the quality of the material itself. And the other is its fit. And they're both really important. And so you could wear a good 3-ply cotton cloth mask, but if it's allowing things to flow in and out the sides, it's not going to work very well. You could wear a surgical mask, which are pretty easy to get now and they're pretty cheap. Those have very good quality. Same thing, they tend to have some flow out the sides. So I saw that Fauci, for instance, was recommending to wear like a surgical mask and then wear a fairly good fitting cloth mask over it, which you can adjust. Cloth masks you can adjust and play with and sew or whatever. And that can help with the fit. And that's how you achieve actually very close to N95, meaning the N95 mask is sort of the gold standard, gives 95% filtering capability. It eliminates all particles down to 0.3 microns, which are the aerosols included, at 95% if it's well fitting. And with this double mask that I just described, which is much cheaper and easier to get, you can reach as high as 92.5%. And so that's why those are being recommended.
So we've got, if people are wearing double masks, And is it safe for them to gather indoors, or in that case, if you're gathering with people indoors, everyone's wearing a mask, make sure all the windows are open as well,
Yes.
or don't gather at all?
That's
What's your thoughts?
It turns out the ventilation is very important, and ventilation differs in different buildings, and it's hard to predict what you have. At home, ventilation is not the greatest. Typically, in office buildings, not the greatest, partly because they recirculate the air a lot, and that allows aerosols to accumulate. They do that to save money in heating and air conditioning or whatever. So indoors, it's certainly better with good masks. You're better protected, but you can still, over a long period of time with a lot of people in a room, have a significant accumulation. So it's important, like I said, situational awareness.
Mhm.
We've all been in situations where we're in a room, we're talking to a lot of people, and you feel the room get stuffy. You know what I mean? We've all had this. That's a very bad sign in this circumstance. Get outdoors, get the windows open, reduce the density. Always be aware of these things.
So kind of looking toward the future, with last month's holiday surge subsiding, are we past the worst for the year? There have been predictions of a springtime surge, and so maybe you can talk about what would cause that. And can we expect things to start getting better as we get closer to summer?
So I can start that. And I know Scott and I have been looking at this and reading a lot about a lot of articles. And it's related to this issue of masking and distancing and continuing to mitigate. People are getting tired of this pandemic. And there's a phenomenon called pandemic fatigue. And that is something that's been hitting people. And as the case loads drop, people let down their guards. So right now, this week, if you look, the US case rate has stopped falling and is starting to turn up a little bit. We'd like to recommend that everyone not let down their guard. We're approaching a period where, with the vaccine, if we continue to work well, I think we could really get things under control, really limit the deaths, and really more quickly reach a point where we're back to something much closer to normal. And so it's very important that people right now really stick with the discipline. I know it's hard. We're all tired of it, but this is the time to do it. And I think that we will see things improve tremendously. I'm always with the caveat that there could be some variant that causes us a lot of trouble, but so far it looks okay. I don't know if others have anything to add to that.
And so the springtime surge that has been predicted is more connected to our behavior than to something else, to variants or specifically to variants or something else.
I think it's probably a mixture of both. It's not an either or. We have both things going on, but the surge we had that we just experienced, most of that was probably both in the US and UK, was mostly dominated by pandemic fatigue more than variants. At least that's what the models suggest. But each surge will be a little bit different, I think, and we can think of it as a mixture of the two. In either case, the strategy is the same. Better masking, better distancing, great care in our ventilation.
And washing hands.
Making predictions are hard, especially when they're about the future. There's so many variables here, and human behavior is hard to predict. It's hard to say, but we know for sure that if people mask, distance, keep in ventilated spaces, we're going to keep this to a minimum. There's just no doubt about it. And get your vaccine when you have the opportunity.
So before we move to the Q&A part of our discussions, anybody have anything you want to add regarding vaccines that you wanted to touch on that maybe we didn't have a chance to yet? Great, we've got a lot.
Andrea, could I just make one point there that's very important? I started talking, but I was muted.
Oh.
One thing that's very important for people who are not immunologists to know is that when you get your vaccine, you're not good to go right at that point. Okay, so if you're taking one of the 2 vaccine formats, the Moderna or the Pfizer, you really need about 2 weeks after the second vaccine to get the full immune effect to kick in. And with the J&J, I would guess, Caroline or Chuck or Scott could correct me on this, I would guess about 3 weeks probably after the vaccine for your immune system to protect you. So that's a very common misconception that I get, that getting the injection is not a get out of jail pass.
And another reason why that the physical distancing and mask wearing continues to be important.
Absolutely.
So we can move on to the Q&A part of the discussion with questions that have been submitted by our attendees. And I invite all of you, any of you on the panel, to weigh in on any of the responses. So the first question, why is it important for everyone who can to get a COVID-19 vaccine? Scott, you want to start that one?
Sure. You can think about vaccines 2 ways. One is at the personal level and one is at the population level. Most of us think about ourselves or our families first, and whether we think this is safe for our family or whether this will provide sufficient, protection to the self. But ultimately, we're community members, and for a vaccine to really do its job, you want the whole community involved, and that protects everybody. And it's really a greater investment for everyone for the future. So there's really no other way to think about it than I think at the community level.
Well, as Scott alluded to, I mean, basically what Scott's saying is that we need to achieve what's termed herd immunity, and for a good outcome. And that, for COVID-2 is what, Scott, in the order of 70 to 80%?
Probably, yeah.
Yeah.
We'll know when we get there.
Either by immunization or infection. And given the outcome that you can have by infection, I mean, this week we're over, 500,000 deaths in the US. And so we certainly want to achieve this number of protection, 70 to 80%, by immunization and not infection. So.
And
so
Andrea, another point that I wanna do before we move to the next question is that these vaccines are performing really well, and that's something that we have to remember. Is their efficacy is great, their safety is looking very good, and is looking even better as more days go by. So it is really encouraging everything that we're seeing. So again, to protect yourself, to protect your loved ones, to protect others. These are some of the best vaccines that we have out there. So if you have the chance, get it.
So, and kind of continuing on that point, another question that we have is, once you're vaccinated, should you continue to get tested for COVID, or will you test positive after the vaccination? Is testing, does it become unnecessary? What's the answer to that?
I'll start that one. Getting the vaccine will not affect your test result. They work differently. And I think while we're still working on the vaccine here, I think getting continual testing would be a good thing to do. Because there, may be some very small percentage. There is some very small percentage. It's not 100% vaccine. Mm-hmm. And you'd like to know about it if you're one of those very rare people who gets it. People who've been vaccinated and do show up positive afterwards have very mild symptoms to being asymptomatic. So that's just another way to look at how well, that's another way to say what we were saying earlier, is that the deaths and the hospitalizations go almost to zero with the vaccination. But I think testing is still an important thing to do. It's quick, it's easy, and it's working and it's readily available these days, finally, certainly to the campus community.
Right.
Good-
And
can I ask a
another question.
I wanted to ask you this follow-up.
Oh, go ahead.
Stuart. So that, you know, if you test positive, that means you have virus and you're shedding. So that also gives us some understanding, if we test regularly people who have been vaccinated, to the extent to which they could be contributing to the transmission, right?
Yeah.
Absolutely. Yeah.
Yes.
So another question related to the vaccines, kind of a dual question. Can the mRNA vaccine cause a dangerous immune overreaction in the body? And also, are there specific concerns regarding the vaccines for individuals who have immunocompromising diseases? Is one type of vaccine better than another?
I can take that. There's enormous variability in the degree to which your immune system reacts to the vaccine. We've seen that already. Some people really get floored from it, others hardly feel anything. And that's just normal variation in how the immune system is recognizing this. And it's probably based on your prior history with viruses and so forth. In terms of patient groups, we're not going to recommend provide specific medical recommendations, but in general, there's been no contraindication to suggest we shouldn't use the vaccines in any group. And that includes patients with cancer, that includes patients with immunodeficiencies. Most of them, the risk of COVID far outweighs any risk from the vaccines. And that's been held up now with, you know, the 45 million people who have been vaccinated so far.
And going along with folks who are vaccinated, an audience member asks, says, our daughter lives in Spain and has been vaccinated. If we also are vaccinated, should we have concerns about her visiting this summer?
Well, that's a great question. This almost so you can look at this from a cold hard risk standpoint and ask, you know, what is the risk of her bringing it to you or vice versa? And if everyone's been vaccinated, the fact of the matter is the risks, the joint probabilities of infection are very low. And you have to trade that off against the value of being with loved ones and the benefits that provides. So I think those are the things to work through in your own mind.
Yeah. Any other comments from anyone else?
You're saying
I guess. Go ahead, Joe.
I was just going to say, it's you know, you've reduced the probabilities tremendously by about a factor of 100 if everyone's been vaccinated. So you're getting back pretty close to normal. There are still, you know, you often have, you know, well, that small fraction who could still get sick, but even there, we don't think the consequences are so great. So I would think it was pretty safe.
And what if it were the other way around and the parents are vaccinated, their daughter's vaccinated, and they want to go visit her in Spain? Same?
I think the virus doesn't really care what country you're in.
Yeah, yeah.
Yeah.
So, Joe, here's a question, probably in your area, and Stu as well. Someone's asking about takeout food, the safety of takeout food. I reheat hot items, but what about cold food? And related to that, should we be wiping down our groceries with disinfectant?
Well, I do. I'm very careful about cold food coming into the house. It looks, I mean, in the very beginning of this, the emphasis was on what are called fomites, droplets on materials, you know, picking things up that people have done it. And I suspect that that's still a real transmission pathway, but I think indications are now that it's small compared to the airborne component. So I never assume it's gone. I personally take care for these things, and I reheat food. But maybe, I don't know, Scott's been following the journals. Maybe there's some more information on this now. I don't know.
It's hard to find evidence of outbreak of any kind from the food itself. It's usually human to human transmission, either due to the ventilation in the environment, or you've got a bunch of line cooks working in close contact with each other. Things that, almost every outbreak we've seen has been aerosols from person to person. So.
Yeah, I would agree with what Scott just says. You can all, especially in the beginning of all this, nobody knew what was dangerous and what wasn't dangerous. So we all erred on the side of caution and sprayed down all our groceries. And I think that with the passage of time, this person-to-person aerosol transmission is seeming like the most common. It doesn't mean you can't get something from a surface. But I think it's just a much lower probability. I don't think it hurts when we bring the groceries in, we lay them on a table, I spray them with alcohol and come back 10 minutes later and put them away.
Yeah, essentially that. It's much more probability. I think that's clear. But there are cases that, where people had no contacts. And so they're rare, but there are documented cases. So I would not call it zero. And until we know better, I'd rather err on the safe side personally.
Yeah. Sure. And most of these things we're talking about are not particularly onerous to do. They're easy. So why not do them is how, for me and my family, that's how we approach it.
Yeah, and Andrew, I think that this is also that, like a lot of a personal choice, right? How do you tolerate some risks? How do you interpret some of these things? But one thing that I want to remind everybody is, like, even if you disinfect everything, even if you reheat, we know that one of the best lines of defenses is washing our hands, right? So if you bring your groceries and clean them, wash your hands. And as soon as you come from the supermarket, wash your hands. If you get some takeout and grab the box, wash your hands. So, the first contact that we have with our face is our hands. So if we have and we keep very good hand hygiene, we also decrease the risk of transmission significantly. So that's something important to remember.
Yeah, and a really easy practice.
And it's one of these things that could depend a bit on age, because keep in mind that every year older someone is, the probability of a severe consequence of getting COVID is about 10% higher. So every 10 years, it's a factor of 300% higher. And so I think for older people, the additional precautions are worth much more. And that's also something you could factor in.
Well, the fact that washing hands and, social distancing to avoid air exchange is important is illustrated also in the fact that influenza this year, the influenza activity so far as I'm aware, has been greatly reduced. So these changes in behavior are having a broader positive effect.
Yeah, there's a report that just came out of UC Davis Medical Center. They've had 0 hospitalizations from influenza this year.
Yeah, pretty remarkable.
That's really amazing.
Excuse me. So I think we have time for one more question. And this, again, from one of our audience members. If you tested positive for COVID-19 and your body's defense system worked well, meaning you didn't develop symptoms, why should you get the vaccine?
Well, I'll start that, and Scott, this is more medical, you may have more to add, but that doesn't necessarily mean, first of all, you can get it again. But to me, it seems like if you've gotten it once, then you could maybe call that like your first shot. And then you would get another shot, and that would be the equivalent of the booster. And that's really going to amp up your immune system. So I would imagine then you would be extra well protected. And what we don't know yet is we don't know, either for the vaccine or for a regular infection, we don't know how long that immunity is going to last. There's a lot of data out there, but there's not a clear consensus, to my knowledge, on that point yet.
Yeah, and that's been the CDC's recommendation. If you've had it, get a single dose of Moderna or Pfizer. Chuck, I don't know if you have any thoughts on this as well.
No, not beyond what the two of you have mentioned.
So the bottom line is get your vaccine when you can.
Yeah.
Well, that bottom line is immensely important, actually.
Well, great. So that about wraps things up. Any other final comments from anyone?
I think it might be good to just mention or encourage everyone to be patient. I know everyone is ready to go today. And the way vaccines are getting rolled out, that's evolving across the whole state. The good news is production is really scaling up quite a bit right now for both Pfizer and Moderna, and with the Johnson & Johnson one almost certainly being approved tomorrow. We'll see that wave coming in another month or so. So everybody, I think, just needs to really hang in there and for all age groups, adolescent and up.
Yeah, absolutely. Absolutely. Well, I want to thank everyone for their really excellent questions. Many of those we received over the past few days actually helped shape the discussion portion of the presentation. Obviously, we weren't able to get to all of the questions, but we'll address as many as we can in the follow-up article that will be published on the Current. And a recording will be available through the Current. Thanks again to our panelists, Professors Carolina Arias, Stu Feinstein, Scott Grafton, Joey Candela, and Chuck Samuel for sharing their time and their expertise. Bye, everyone.